can start, I think. Hi, everybody. Good morning. And welcome you to the panel discussion on advanced research computing applications. So we have speakers. I'm just naming them right now. Those are Nannan, Karakoc, Seyoon, Victoria Rolandi, Olivia Sanderfoot, and Fangming. Just a request that before we start the program, just a request, if you are not talking, please keep yourself mute. And if you have any question or feedback or comment, just raise your hand and we will allow you as situation permits. So want to thank you all our speakers and the participants. Hope you all enjoy this. So what I will do is maybe I will ask each of our speakers to tell us about themselves and what kind of research they are involved in, what kind of computation and computational applications they are using. And then once that round is over, then we will come to question and answers. So let me start with Nannan.
Okay, let me share my screen. Good morning, everyone. I'm Nannan from the Department of Ecology and Evolutionary Biology. Today, I would like to give an example of the advanced research community application. It's a global research on biodiversity. It's based on huge community science data and Huffman. The community science data is a IDRE data. First, I would like to talk what we want to do and why we want to do the urban biodiversity research to see if it have a scaling effect, because it's important to get the scaling effect to make a policy for cities to do better management. For the research, previous research, there are some contradictions, like the F. He said that cities are hotspots for threatened species. But the NAP said the urbanization is increasing faster than ever. It contributes to a global extinction process. Then the Fatrini said even species area relationship, it's index that towns are not different from the natural area. And Callahan has said his strongly support for the long-standing spatial area relationship. They have different sayings about the urban biodiversity. What should we use to do the policies to better manage urban? Are there samples large enough before that we can say this studies they based on countries like Australia, Germany, Italy, and America? They're not global research. Maybe their data are not large enough to get a conclusion that cannot be used globally. Likely now we have the community science data, the IDRE data. It's huge. It covers 253 countries and regions. Now we can use this huge data, and we're wondering that's the well-established the species richness and area relationship when it's log transformed, it's a linear relationship. We are wondering that if we get such huge data, are they in urban area, do they still have just a linear relationship? Can it be non-linear? So I do a research that based on the mixed effect generalized additive model. It's have no assumption that it has to be linear. It can be untrained, just according to the data. And our last question is we want to see the effect of urbanization. And this is the sampling we get. It's actually much more than previous research. It covers 26 and 2,603 cities and 1616 nature reserves. The gray one is the urban area without the threatened species. The red one is urban area with threatened species, and the green ones and the nature reserves. And the data we used beside the sampling map in the last slide is IDRE data. It not have more than 75 million complete checklists and the urban area, because we want to see the effect of urbanization. So we can't use the administrative boundary. We have to use the real urban boundary. So we use the urban boundary data that based on the satellite image data and other data, and we use the grid population of the world data from the NASA website. Other than the urban area, we want to do comparison to the nature area, nature reserves, to see the urbanization effect. We also got the globally protected areas to do the research. How did we process that huge data? We have to say thanks to the Huffman tool, because it's very large. It's compressed the IDRE data. It's 138 GB. And we upload it to Huffman, split it year by year, then limited to area and nature reserves, and set criteria to remove bias like the spatial bias, because it's a community science data. So it's observers, they are not all the experts, they can be anyone. So they have the sampling bias and the spatial bias, many bias. So we have to set criteria to remove such bias and remove the duplicated records and roll up the data to the species level, because the data, when they submit, it can be subspecies and can be a level that upper than the species level. So we have to roll up to the same level to do analysis. The last step is calculate the species richness we want, and other index like the in cities, which species it can cover many cities, which species only exist in one cities. To do all the steps, we use based on the Huffman and plus other software like the ArcMap and like R and the Unix command to do it. For me, the Huffman advantage, the first one is it can store data. If you have big data, you can upload to the Scratch. It have large space. Then you can deal with your data easily. Another is submit your jobs to let Huffman to deal with your data for you, even you are asleep. Actually, there's another advantage that Huffman, when you collect the files on Huffman, it respond very faster than your computer, like the Office software. You can check your data to see if there are issues very fast. And this is what we got. Actually, our assumption is reasonable. It's in urban area, it's really not a linear relationship as a well-established power law. It's clearly that it exists two phases, that when the urban area is not large enough, it's the slope is much steeper. But when the urban area is large enough, it's the slope go very slow, that you can see the distance from the nature to the urban area. It's become larger and larger. When the urban area is large, that it really have a very deep impact on the seriousness.
Nannan, could you please wrap it up so that we could go around the other speakers as well?
Yeah. This is the last slide. Okay.
Sorry about that. Thank you.
Yeah, and the last image it shows that the urban area is when it's large, it's really a hot spot for the threatened species. It's surprising. Thanks, that's all.
Thank you. So the next we have is El.
Hello. I think I have to share my screen. Yes.
Yes, you can.
You can see, right? Okay.
Yes.
Okay. Wait a second. Okay. So, hello everyone. My name is Karakoc, and I hope you can see the slide. Is everything fine? Can you see?
Yes.
Okay. Well, I will briefly talk about challenges, requirements, and opportunities for 3D in silico investigations. I'll try to keep it fast and short. Well, as many of you may know, 3D in silico investigations are physics-based computational models focusing on providing meaningful and predictive assessments for hemodynamics, tissue mechanics, and physiology in healthy and diseased states of organs. Well, the in silico models and simulations were originated to assist clinicians and surgeons in choosing optimized operational plans for patients, especially in recent years with the increasing know-how, collaboration, and democratization of computational tools. These models became capable of providing more predictive and more personalized treatment plans with possibilities in patient time reductions in the operating room. Likewise, these models and simulations became a routine strategy so that medical device design could be accelerated with virtual testing the proposed designs before they are deployed in animal or clinical studies. Hence, in silico investigations with their predictive outcomes could support clinical decision-making. In addition, the regulatory processes for biomedical devices are also benefiting from the in silico results while minimizing the animal tests and clinical trials. Well, of course, in order to create biomedically approvable evidence, computational requirements must reach to a strict level of precision modeling and highly accurate results. Therefore, research organizations and academic units have been in need of multidisciplinary collaboration, experienced staff, and training activities for junior researchers for the targeted research fields. And in addition to these, of course, high performance computing and data management are very critical. So luckily we have Hoffman cluster, which works very nicely for our simulations. And there are critical core technologies in the in silico investigation, such as image processing, data segmentation, and domain reconstruction that are basically used for hemodynamic simulations and fluid structure interactions. Well, we are very lucky that data democratization and open science initiatives for biomedical computing and digitalization have been increasing day by day. So as a result of this very strong culture of collaboration and sharing has been also established across the disciplines and organizations. So now we have a variety of open source pre-processing, processing, and post-processing packages to overcome the challenges within a growing international ecosystem of academia, governmental agencies, and industry. For instance, we have various platforms such as ITK Snap, 3D Slicer, the Vascular Modeling Toolkit, VMTK, which provides functionalities not only for image processing, but also have other supporting tools and plugins, such as Medical Open Network for Artificial Intelligence, MONAI, computer graphic algorithm libraries, and open cascade technologies, to name a few, which greatly contributes to automatic segmentation, labeling, and mesh generation. Of course, in addition to these, nowadays, a complete pipeline from anatomic model construction to numerical simulation and analysis are also possible with the open source software suites, including SimVascular, Crimson, FE Bio, which are created in different universities across the nation. And there are also more general purpose solvers such as OpenFOAM and Phoenix that are also used for this type of simulations and fluid structure interaction problems. Well, of course, I mean, there are challenges as in many different fields of research. So in terms of the future challenges, we have to provide high fidelity parameter inputs, such as high resolution of clinical imaging modalities to create patient specific simulations accurately. And also more careful collaborations with engineers, medical doctors, and soft tissue vascular biologists are very important for development of specific material models based on mechanobiological processes. And also requirement for real-time results with reduced order modeling and machine learning algorithms are also very important without renouncing the accuracy. And well, I think the most important, well, let's say one of the most important things is to gain community trust with reliable and computationally efficient results, which will pave the way towards virtual clinical trials for FDA approval and create new pathways for the development of, well, medical devices and treatment methods for using the patient-specific data. Well, I just briefly, talk about the possibilities, challenges in the field, and I would like to thank you all for listening. And I'll be happy to get your valuable feedback about this multidisciplinary field. Okay, thank you very much.
Thank you.
I think I will stop sharing. Yeah, okay. Thank you very much.
So next speaker is Seyoon Koo.
Hello, my name is Seyoon. I'm a postdoc in computational medicine, working with Dr. Hua and Ken Lang. I'll talk about the Open Mender project, which includes any software for statistical genetics. For the field of genetics, the pipeline for processing can be very long and be very complicated, involving up to dozens of tools. For coding haplotypes, there is a tool named GATK, and for manipulation of the SNP data, standing for single nucleotide polymorphism, there's a tool named Plink. And for phasing them, and because there are missing data, for imputing the values, there are tools like Impute, Beagle, FastPhase, and Mendel Impute. And for heritability analysis, there are tools like GATK, Mendel, and there are many methods that has been proposed for association tests. And there are many tools for each of the tests, like Plink, FastLMM, Sage, Gamma, and others. And for linkage analysis, there are tools like Merlin, Solar, and Sage, and those are mainly written in languages like C++, C, or Fortran, and they are often very hard to be modified and suited for very specific uses of each new researcher. And for statistical analysis and visualization, there are other softwares like R, MATLAB, and others. And for easier to use interfaces, people might write glue codes in Python, Ruby, R, or other languages. So it is very scattered set of software in this field. In a dream world, in the ideal world, one would hope for a unified programming environment that is efficient and eases new method development, fosters scientific collaboration, and encourages reproducible research. And that is the goal of the Open Mendel project. It is statistical genetics toolbox in Julia, targeting accessible computing with large-scale datasets. It is successor of Dr. Ken Lang's Mendel project, whose motto is to be Swiss Army knife of genetic analysis programs, and it includes over 30 possible options of analysis, 30 possible pipelines of analysis. This project started over 30 years ago, and at the time, the most popular programming language for computational mathematicians was Fortran. So it was naturally written in Fortran, but unfortunately, it is very verbose language, and it is hard to bring in people to collaborate. So a decision was made to use a new programming language, Julia. It is free, easy to install, it is cross-platform, and it is easy to learn because it is high-level language like Python or R. And it has clear syntax that lends itself to compact and readable code, which is more important to computational statisticians. It is fast, and the code is easily parallelized. High-performance computing techniques can be relatively easily involved than using other languages, and it is easy, and it has modern package management system, where modern package management system means nicely separated versioning of packages in each separate environment, which is not included by default in languages created in 1990s like R or Python. And it has many statistical analysis libraries, and it is approaching to the level of R and MATLAB. So what is being done for the project is development of scalable statistical methods for biobank scale genetic data. By the biobank scale, we mean up to the size of million by million, and it uses dimension reduction techniques and as well as parallel computing techniques involving graphics processing units and multi-threading. The project like OpenNet mixture for inferring global ancestry that I presented earlier this month is one example of those packages. And other application would be genome-wide associated studies for longitudinal data, or for data with ordinal categorical phenotypes, or joint analysis for multiple phenotypes at the time. And as well as that, the options for original Mendel package are all transfers to this set of packages, including imputation and phasing, aim selection, ancestry informative marker selection, linkage analysis, heritability analysis, and others. And another lane of emphasis is development of core input-output packages for various genetic data formats like PLINKS, BED format, Oxford Visual format, PJ format, VCF formats, and others, fostering direct mathematical operations on fixed width data format, like Plink BED format. And it can use multi-threading or use GPU. And we keep packages in the form that is more accessible by open sourcing on GitHub with nice documentation and unit testing. And we also work with execution of developed methods for large-scale experiments in terms of both programming and utilization of resources. For example, large-scale genome-wide association studies with millions of SNPs. It is accelerated using distributed environment on like Hoffman 2, and we provide ways how to do that in an easy way, and because not all datasets are suitable for Hoffman 2, we also, because there are contract restricted datasets, we also use cloud services like Amazon Web Services to keep the security of access to dataset. Thank you. TV, you're muted.
Thank you, Seyoon. The next one is Olivia. Victoria, sorry. I'm sorry.
Okay. Share the screen. Can you see my slides?
Yes.
Okay, so hello everyone. My name is Victoria Rolandi. I'm a postdoctoral researcher at the Mechanical and Aerospace Engineering Department, and I'm working with Professor Taira. For this panel discussion on the advanced research computing application, I will introduce the importance of ARC in the fluid mechanics context. So for this presentation, for the first short presentation, I first introduce how do we simulate the fluid motion, then why simulating turbulent flows is so expensive, and how having access to high computational resources has opened the door to more sophisticated analysis like flow control of high turbulent flows. So how do we simulate the flows? Actually, we numerically solve the Navier-Stokes equations, which are partial differential equations characterized by a nonlinear dynamics and that can span over a wide range of spatial and temporal scales. How much the simulation costs, of course, it depends on the time step discretization and the spatial discretization, as we can see here in this picture. The main step of the correct choose of the discretization for a proper simulation is the need of capturing all the spatial and temporal scales of the flow. For turbulent flows, this choice is based on the concept of the energy cascade. There is a constant transfer of energy from large eddies, from large scales to smaller and smaller ones. Then these small scales are dissipated through viscosity. The important point is that we have to capture these small scales, so the length of these small scales is what determines the discretization. For turbulent flows, increasing the Reynolds number, so increasing the turbulence, the temporal scales become shorter and the length scales become smaller. That's why it's so expensive to simulate high turbulent flows. However, we are very interested in simulating such flows because these flows are the ones that we find in the most of the engineering applications. Fortunately, in the last years, the increase of the computational resources has given access to this simulation, so we are now able to obtain high fidelity flow field data like these ones showed in these pictures. Why this is such a big step? Because now, having access to this high fidelity flow field data, we can start other analyses at high Reynolds number, like flow control, that are very helpful or useful for example, the design of the next generation vehicle. For flow control, what we need is to extract the physical insight from this data, and we can do so through the so-called resolvent analysis. The resolvent analysis tells us how the flow responds to an harmonic input of the system. So we have the Navier-Stokes system and the input-output relationship. To analyze this input-output relationship, we need to perform a singular value decomposition of matrix A. What we need to understand is that for turbulent flows, the number of degrees of freedom is very huge. We can reach the order of 10⁸, 10⁹. So it means that this vector, the number of entries of these vectors, is of this order, but in particular that this matrix A, is the square as a number of entries, which is the square of this value. So we can understand that the dimension of the discrete operator is extremely large. This is why the applications of resolvent analysis have been restricted to simple laminar flows or flows at moderate Reynolds number. First of all, because before we didn't even have access to high fidelity data at such a high Reynolds number. But now that we have access to this data, we still have problems in terms of computational calculation and memory allocation to perform these analyses. So how to extend the resolvent analysis to highly turbulent flows? There are, the last years, there are mainly two ways that have been taken. For example, the randomized resolvent analysis that computes the singular value decomposition in a low rank approximation matrix, or hierarchical decomposition, which takes into account that the entries of the matrix are, most of the entries of the matrix are 0. I will not enter now in the details of these two ways, and I will talk about them more in details in my next seminar, which will take place at the end of April. So for now, what we can see is that we have seen that how little brain flows are important for engineering applications, and that the increase of computing resources has opened the door for these useful and more sophisticated analysis. And but however, we need to extend the resolvence analysis to high turbulent flows, so that we will be able to analyze and find new ways to design the next generation vehicle. So thank you very much. And if you have questions, we'll talk later.
Thank you, Victoria. The next speaker we have is Olivia Sanderfoot.
Hi, everyone. Okay, I'm pulling up my slides now. Can everybody see my title slide?
Yes.
Fantastic.
Yes.
Hi, for those who don't know me, my name is Dr. Olivia Sanderfoot, and I am also in the Department of Ecology and Evolutionary Biology. I am a postdoc in Dr. Morgan Tangley's lab, which focuses on global change ecology. And today I'd like to share a bit about my research integrating big data in ecology and atmospheric science to support bird conservation in the age of mega fires. So, as many of us know, birds face many threats, including overexploitation, habitat loss, and environmental pollution. Air pollution specifically is a known risk to birds and other wildlife, but to date, air quality has often been overlooked in conservation biology and wildlife management. I am particularly interested in thinking about how one pollutant, fine particulate matter, impacts birds. Fine particulate matter, or PM2.5, includes all suspended liquid and solid particles that are less than 2.5 microns in diameter. So this is the stuff in the air that's thinner than the width of a human hair, really, really fine. And because it's so fine, it can become deeply embedded in our respiratory systems upon inhalation exposure. There are both biogenic and anthropogenic sources of this pollutant. Biogenic sources include windblown dust and sea spray. Anthropogenic sources include vehicle exhaust and industrial emissions. Fine particulate matter is a major risk to public health. It has been linked to cardiovascular and respiratory disease, as well as thousands of premature mortalities every year. But because it's such a major risk to public health, we actually have really, really good data on exposure. And our estimates of PM2.5 come from ground-based monitors, air quality models, and even satellite instruments. So this provides a really exciting opportunity to think about how PM2.5 may be impacting the health, behavior, distributions, and population trends of birds. I think it's especially important that we focus on this pollutant, not just because it's already been shown to have major consequences for human health, but because we're actually seeing PM2.5 increase in many parts of the world directly as a result of increased wildfire activity. Climate change and a legacy of fire suppression is increasing the frequency, intensity, and severity of fires around the world, including here in the United States. And because of this, we're seeing many large, fast-moving, difficult to control mega fires, which are these big fires larger than 10,000 acres. And we're even seeing fire in places that were once entirely free of flames. And where there's fire, there's smoke. So not only are we seeing more fires on the landscape, we're actually seeing more smoke on the landscape. And we don't know what the consequences of this might be for birds and other wildlife. So going back to that initial slide, there are lots of things that birds are facing that are posing risks to their long-term population trends, and smoke might be one of them, but this has not been explored in the scientific literature at all. We know that these smoke events can be quite disastrous. This is a true color satellite image of the Western United States taken on September 9th, 2020. The orange dots represent fires and thermal anomalies. And you can see from all of these orange dots are incredibly lengthy and intense smoke plumes stretching out hundreds of miles over the Pacific Ocean. So this is just one day what smoke exposure looked like in the Western US during a very intense fire season that took place in 2020. And you can imagine that if you were a little bird flying through this might seem very stressful and could have all sorts of repercussions for health and behavior. So that this is really the motivation behind my work. Like I said, we don't know that much about how animals are impacted by air pollution, but we do know that birds and other animals just like us are certainly exposed to air pollution. These are some photos of wildlife taken on camera traps as part of a study that I was part of in Washington State on smoky days. And you can see that birds and other animals are certainly out there in smoke. They have to forage, they have to find food. So exposure is definitely going to be part of their day-to-day life during the fire season. I recently led a literature review to synthesize research to date on the impacts of wildfire smoke on the health and behavior of wildlife, and we compiled all of our findings in this conceptual model, which really sets the stage for this research agenda that I'm hoping to pursue as part of my postdoc, and hopefully in the future as my own, as when I run my own lab. So the idea is that wildfire smoke contains both gaseous and particulate pollutants, and these pollutants lead to health effects. But wildfire smoke could also lead to short-term habitat changes or shifts in the visual and olfactory cues that animals use to navigate in their environment that could trigger behavioral responses. And both these health effects and behavioral responses could scale up to influence vital rates, including survival, reproductive success, and growth and development, and in turn lead to population level impacts. And if anybody has questions about this chart, I know it's a little bit complicated, I'd be happy to dive into it in the Q&A or provide a link to my open access paper so you can read more about it. So our review really identified some crucial knowledge gaps. So first of all, we have very limited inference on the impacts of wild free-living animals and the sensitivity of responses to specific pollutants. So what this really means is that while I can make some general assumptions about what the possible consequences of smoke exposure are for birds, I really don't have an idea of how those responses vary linearly as a direct relationship with specific concentrations of pollutants that are contained in smoke, such as carbon monoxide or PM2.5. And this is really what we need in order to estimate dose response relationships for those vital rates, including survival, growth, and reproduction, as these are the parameters that ultimately determine whether a population increases in size, decreases in size, or is in fact stable. We also need to characterize behavioral responses to smoke because ultimately our observations of birds are also dependent on how they're acting and what they're doing. So if we don't really know how smoke just impacts their behavior, we're not accounting for that in our models. I am particularly interested in thinking about movement and migration, as some recent studies have come out showing that smoke could have pretty big impacts on how birds navigate through space and time. So, whirlwind overview of these knowledge gaps. How are we going to answer this question? We're going to use high performance computing. So there is amazing data in atmospheric science to think about exposure to smoke. As I mentioned, we have data from ground-based measurements, satellite data, and air quality models. And these air quality models may be examples of dispersion modeling, of photochemical modeling. There are literally dozens and dozens of air quality models that have their own pros, cons, and nuances to consider. And so we have this really high resolution spatial and temporal data on how air pollution may be affecting bird communities, but we have not really linked those exposure estimates to observations of birds. So how do we think about where birds are and what they're doing? Well, we have lots of tools in ecology to study where birds are and how they behave. We conduct surveys, we put out trail cameras, we have bioacoustic recorders and GPS and radio telemetry, and we even use non-invasive DNA to track animal populations. But to date, these data sets have never been combined to really think critically about how smoke exposure is impacting spatial and temporal distributions of birds. And I think a tool in ecology that's particularly exciting is something that Nannan talked about earlier, community science. Community science is a growing movement and a type of research in which non-professional scientists get involved in collecting data for scientific research. It's really just public participation in science. And that's a really awesome thing, because that means that regardless of your background and your training, you can get involved in collecting valuable data for scientific research. And a great example of this in ecology is eBird, which is a global citizen science database that supports opportunistic, semi-structured data collection. And as Nannan said, there are more than 1 billion observations of birds in this database. So there's tons and tons of data on bird observations from all around the world that we can pull from to think about how air pollution, specifically in the context of wildfire smoke, may be influencing where birds are and population trends over time. And I'd be happy to answer any questions about what I mean by opportunistic, semi-structured data collection. Essentially, birders who participate in eBird get to choose where and when they conduct their surveys, and they collect additional metadata that helps us better understand the detection process and account for some of the biases that Nannan mentioned. So in my current postdoc, I hope to pull in big data from atmospheric science and big data from ecology, specifically bird observations from eBird, to think about how wildfire smoke is influencing the behavior and distribution of birds. My goals are really to assess the potential for smoke to act as an ecological disturbance all on its own, separate from fire, and identify which species may be most vulnerable to this stressor. Ultimately, I would love to determine critical thresholds for exposure that really serve as a trigger for a change in state in bird populations. I think this would be very, very valuable for management in order to think about how to mitigate smoke exposure to protect both people and wildlife. So that was a whirlwind overview. Happy to answer more questions. Quick word of thanks to the National Science Foundation Graduate Research Fellowship Program that funded my PhD, the McIntyre Stennis Cooperative Forestry Research Program that's funded some of my research work, as well as the UCLA Department of Ecology and Evolutionary Biology, the UCLA Global Change Ecology Lab, and of course, IDRE for supporting the current work that I do in the lab. So thank you so much for your time. Feel free to reach out with any questions. And if you have a cool air quality data set that you would like to link to bird observations to think more about air pollution impacts on avifauna, definitely let me know. Thanks so much.
Thank you, Olivia. Next speaker we have is Fangming Z. Could you come online, Fangming?
Sure. Let me share my screen. Play. Okay, can everybody hear me well?
Yes.
Okay. Hi, this is Fangming from the Woman Lab from the Department of Chemistry and Biochemistry. So we are interested in developing a technique that allows us to look into the organization of the mouse brain at the level of single cells with molecular specificity and yet with high throughput. So what do I mean by that? To this audience, we ultimately wanted to know how the brain works, but to this audience, we know about the artificial neural network, which is a mathematical construct that consists of neurons or nodes that each of which does simple operation, linear summation and thresholding. And then when they talk to each other, they can achieve complex tasks. So our brain works fundamentally in a similar way, except that it's a lot more complex, even than the artificial one we built to date. And the human brain consists of more than 100 billion neurons, and each of which is a complex molecular machine, if you will, and they together form a complex operation and organization. So the question is, how do we survey such a complex organization in order to ultimately know how it works? So, well, one can tackle this, the complexity of human neurons, from many different perspectives. So early on, people noticed that different neurons have different shapes and sizes, and the fancy word for that is morphology. So here are two examples of different neurons. They definitely look differently, but later on, people find and they also respond differently to stimuli, and they're located in different spaces in the brain, and they talk to each different sub areas of the brain. But what's deriving all these complexity or diversity of cells are their underlying molecular signature, because ultimately it is the proteins and RNAs that they made that makes the difference, that the phenotypic difference. So molecular diversity underlies all the other diversity of brain cells, and the question is, how can we comprehensively survey these molecular signatures at the scale of the whole brain? So over the last decade, the field has made tremendous progress in surveying the molecular signatures of brain cells, and the state-of-the-art technology, when you compare with Moore's law, is increasing in throughput at a faster rate, both at exponential growth. And so if you look at the throughput, however, if you look at the throughput of the state-of-the-art technology, which is single-cell sequencing, as of now, the best one can survey about 1 million cells, which is a large number, with a single experiment. However, this still doesn't meet our demand of understanding the mouse brain, which consists of 100 times more cells than we can survey, and let alone the human brain, which is even 1,000 times larger than the mouse brain. And this is all about one brain. And if we want to think about any serious study, you need controls and cases to make the statistic robust. And also, if you want to study development and aging and disease, all of these need more samples. So there is a demand of increasing the throughput of current technology that we can measure molecular signatures of cells. So how can we achieve this? And so the well-accepted approach is the following. So you typically just take a brain sample or whatever tissue sample you like. You get their cells individually and measure their full transcriptome. In other words, measure their individual gene expressions. Because there are more than 10,000 genes in the genome, you make more than 10,000 measurements, essentially. And when we get that high-dimensional features for individual cells, we later on run computational methods to reduce its dimensionality to a manageable level, such as using PCA to reduce it to about 20 or so low-dimensional embeddings or features, each of which is an aggregate of many different individual gene expression. And from that low-dimensional space, you can figure out the similarity and differences among cells, and using that to capture the cell type diversities of the brain. So what we propose instead is, instead of doing this individual measurement at first and then computational reduction later, how about what if we can directly measure the low-dimensional embedding, which is an aggregate signal of many different genes, directly with experiments? If we can do that, we make a shortcut. Instead of having to measure individual genes of 10,000 genes or so, we can measure 20 or so aggregated signals from combinations of genes, and we can do, and hopefully with those features, we still capture the cell type diversity pretty well. So we figured out a way of doing this. Essentially, we implement the matrix multiplication in situ using the chemicals. And there are other benefits by measuring aggregated signal versus individual gene. It just makes the entire experimental workflow a lot easier. That allows us to increase the throughput by a lot. So at the scale of the whole brain, essentially. So it's one thing to talk about the idea, it's another to implement it. So how it works, we're still in the preliminary stage. It turns out the experiment itself is very challenging, but we have some preliminary data to show that it kind of works. So what I'm showing you is a coronal section, a section of the mouse brain, and stained by aggregated signals. And so I'm showing you the same sample measured by 24 different signals, each of which is a combination of many gene expression signals. And you can see that there are distinct patterns of the brain recapitulate some of the known biologies we already know about the brain architecture and the cell types. So without going much into the detail, let me stop here. And we are interested in further developing this technology that combines the computational insight with the experimental design that makes the measurement of molecular signature ultra high throughput, so that we can finally bridge the gap between what we need to access the complexity of the mouse brain or eventually human brain versus with a technology that allows us to get there. With that, I would like to discuss more about it with anyone, and feel free to ask questions about things.
Thank you, Fangming. So I think let's open for the question and answer from the audiences. If you have any questions, raise your hand. I will, and we can go that way. Lisa?
Thank you also very much for those presentations. It's really amazing to see the breadth of research represented by the ECR group, and it's wonderful. So my question is just simply to all of you. So many of you used the Hoffman-2 cluster and continue to use HPC. If you could make one change to the cluster and how it works, what change would that be?
I will defer to my colleagues on that question. I think that's a great question. I actually am in the very, very first stages of my project. So everything's planned out, everything's ready to go, but I haven't run anything yet. So I'm not the best person to answer it, but I think that's a great question. I'm super curious to hear what others have to say.
Fair enough.
I'm in the same case. I will just start my postdoc these days. So I've just arrived in the States from Europe, and I will use Hoffman soon.
Okay. Well, if any of you have any thoughts, please share them with TV. He can reach out to me as well, and we'd love to hear what you have to say. So thank you.
Go ahead.
Can I expand on that question real quick? There was at least one of you who used, I think, a cloud service beyond Hoffman. Why that decision?
Yes.
Oh, in my case, the data I've been using is not suitable for Hoffman-2 because it is health data. It is important to keep the data security, and Hoffman-2 is does not allow contract protected data on there. So I have to go to other routes for that kind of data. It is not Hoffman-2's fault, I would say.
So do you use that computation through the AWS secure that's like for UCLA or like from the medical side?
Yeah, I use that kind of thing. And if I am to change one thing about Hoffman-2, that would be better support for the language I use, because the language I use is pretty minor here, Julia.
Okay.
Yeah.
But Julia is available on Hoffman-2.
It is available, but the support for it is sometimes outdated.
Oh, okay.
Hi, this is, I'm sorry really to interrupt. This is Charlie Peterson. I actually work on the Hoffman-2 system support team. yeah, and this is very curious, and I really didn't mean to interrupt the flow. And like, I am very interested in, especially working with Julia, because mostly that, we didn't really have people working with Julia. And it's more like, people don't know about that. And I'm actually more, especially if you want to reach out to me and or other people to Hoffman team, we're really open. And I'm still really working on getting people, especially with big data, working people with, Spark, Hadoop, and any other data science platforms. And I really, would be interested in, people's thoughts or something like that. So yeah, feel free to reach out to us. I'm really curious on, how people are using it, because, we want to use the Hoffman 2 cluster to, suit your needs. And sometimes we don't know what you guys are doing. So, but yeah, feel free to reach out. So I'm also very curious. So yeah, thank you.
Sure. Thank you very much.
I guess one feedback I might be able to provide, I don't have much experience with Hoffman 2, but we choose to maintain our own lab server, which is painful to maintain, but still we choose to do it. One point, my impression I seem to got from my colleagues is, so for many of these bioinformatics studies, you need a huge data set that you want to load into the memory and play with them interactively. And it might be hard to request a large memory and play interactively with Hoffman if you need to request a node and it only comes hours later, and then you no longer have the time to go with it. It's going to be a problem, I guess. But I don't have much personal experience with it. I guess that's why we also choose to have our lab server, so it's more flexible to do these kind of research.
Oh yeah, and of course, and like I said, please feel free to reach out, because I really want to, especially know, the needs, so we can actually better suit. Because yeah, I'm really interested in getting, big data and memory intensive platforms into, stuff like this, especially people with, large, amounts of data. So yeah, I mean, like I said, feel free to reach out to us. So I'm more than happy to, at least, see what we can do, and, probably, see what other research we can also expand to. So, but yeah.
Thanks.
Any other? Okay, if not,
I have a question. That I want to know that how often do you clear data on the Scratch? Because it's said online, it's 7 days, but I stored the data much longer than 7 days. Then I thought it won't be cleared, but suddenly it's cleared. So I want to know how often.
I mean, I would probably better say, talk to my bosses about that. But normally we do like to, that is in a sense our policy, just to make sure, things are, presented. But typically, when we do like major upgrades and stuff like that, we will clear out older files if need to be, that are, much older. But we typically also send out a lot of emails and stuff to really make sure that y'all know that when we do these upgrades or removing data, that you have that information well in advance.
Thank you.
Prakashan, you had some question?
Yeah, yes, Stevie. Yeah. Before I ask my question, I will just make a comment on Hoffman 2. In my opinion, Hoffman 2 is the best compute resource for the money. If you do the same thing on Amazon or Azure, it will cost you 10 to 20 times more the money. So that's just a comment. Okay, my question is that maybe to the first speaker, that I saw that you were using R to compute. I mean, most modern data processing applications I see is that Python Pandas and Jupyter Notebook, they are much more powerful than, I mean, my benchmark I did some 10 years ago, I compared the R Python and Julia. Among the Julia was the fastest. And the Julia, another advantage is you can parallelize easily. I mean, Python, you can parallelize it too. I'm not saying that R cannot be made faster. If you compare with the Intel Fortran compiler, could be made faster. But is there any reason why you are not using Python Pandas and Jupyter Notebook? Because it has all kinds of nice capability. And now the modern machine learning, everybody's using. I mean, if you're running TensorFlow or PyTorch, it's by default, it is Jupyter Notebook and Python, right?
I'm going to jump in and help Nannan out here. I hope you don't mind, Nannan. Jump in if you want. R is the language that we use in ecology. That is how we program all our analyses. That is what this discipline uses. And it has been actually very challenging for me to work at the intersection of both ecology and atmospheric science and communicate across different languages and data formats and file types. It's interesting to me that we have such a disciplinary divide in terms of how we program our analyses and even how we store and share data. And I actually think that's something to really think about when we build platforms across disciplines is what are the languages of these fields and how can we build out capacity. Obviously, like I would like to learn Python. That's on my ever-growing to-do list of things to add into my toolkit, because as you said, there's a lot of benefits to using Python. But as of right now, like in order to communicate with any of our colleagues, we're going to save our data in a .csv and we're going to do all of our analysis on R. And all of the packages that have been developed to read in, process, and filter the eBird data that Nan and I are using are exclusively in R and don't they don't interact with the other languages that you mentioned. Nannan, do you have anything to add to that?
Actually, no. Other than R, I think now I use the Unix logic. That's a Unix command. I use the AWK.
The awk. Yeah. Yes, you can do that in Unix, or you can use their build in R too. Yeah, to process it for sure. Unix and R.
Yeah.
Makes sense your answer. I was just curious.
I guess just to add to that comment a little bit from a different perspective, I personally uses Python and Jupyter Notebook the most. But that's just because I come into the field of bioinformatics late. And there's a strong tradition established by the community and statisticians that uses R, and half of the packages and all of the existing ones, a lot of communities are still in R, and that's the language they're most comfortable using. So now the bioinformatics kind of split between the two major things. One group's in R, another about half of people using Python. We are kind of bridging in between, but it's not one or the other yet.
Yeah, another question I have you guys is that nowadays the computing is not just done on Hoffman. I mean, one of the users is using AWS, right? So the computing is moving more to containerization, like Docker containers, so that you can collaborate, you can take from AWS to Microsoft Azure within a matter of few seconds, right? I mean, provided you have accounts, everything, and data is ready. So, I mean, those are the kind of modern computing now we are going. In my area, that's what I do. I mean, it's mostly all containerized. And if you have to collaborate with many people, that's only choose containerization, GitHub, and all those tools. So are you guys using those, taking advantage of those things?
I guess those are more on when you are using some kind of clouds or the environment where containerization or virtualizations are more common.
I'm not the expert in it. By container, do you mean that those like Dockers and virtualing? We kind of, I think now it's typical for the Python community to use the Conda environment to manage our packages. It's the bioinformaticians tools are ever growing, and it's harder and harder to manage. It becomes typical to use those kind of things. And I think for Docker, I'm starting it, and the more and more paper submission now requires you to put your code into a container that's easy to share and stuff. But it's still in its early stages. I think we are not the expert in using it and know the best practice on it.
Yeah, that should be something you guys should look into it. Yeah.
Rose?
I'm done with my questions, Stevie.
Okay, thank you.
I have two questions not that far off what Prakashan was asking. My first question is, are any of you using sort of various databases such as either NoSQL or SQL databases? And then which ones? And then the second question is, what are you using for a code repository to store your code? Are you using Bitbucket, GitHub? And did you know the campus has a enterprise license for GitHub?
I can speak to both those questions. I am not currently using any sort of a database of the types that you mentioned. All of the data that I pull in is primarily processed and housed by the institutions and organizations that are responsible for data management. And then all of the file processing is done in uploading and processing .csvs. And I use GitHub. Our team has primarily used GitHub for data storage and pushing revisions.
Cool.
Yeah,
Anyone
I
else?
guess in my grad school, we use MySQL, I think the old school version, to store genomics data, with which we build some online browsers allows people to view the data directly. Yeah, that's and we're using GitHub for everything. We don't know about the license.
Yeah, we are intensively using GitHub for software repository.
Cool.
I've been using the GitLab instead, but I think now that I'm joining the group at UCLA, I think they probably use GitHub. Yeah.
Is there any other question or comment? If not, let's thank our speakers for wonderful presentations.
Great presentation.
And yeah, and for the participants who joined to encourage all our ECR group. So thank you again, and hope you have a wonderful weekend. Thank you.
And I want to say, Olivia, do we gather somewhere after this meeting?
Yes, fellows, if you want to get lunch and hang out, we're going to Chipotle now at 1 in Westwood. I'll put my number in the chat in case you get lost or can't find us, since we've never met.
Your number in the chat, because as you can see, I'm home. I'm still home, and I don't have the car because I arrived 2 days ago. But we will arrive, okay, soon.
Sounds good. Sounds good. Yeah, I'll put my number in the chat and I'll also email it out. I hope to see folks there. Thank you everyone for your presentations and your time. I learned so much. It's really quite an honor to be part of this cohort of such brilliant young people. So thank you for your time.
Yeah,
thank you. And by the way, unfortunately, I am not at the campus, but sometimes when I am and we have some event like this, definitely I'll like to join. So thank you and have a great time.
Thank you.
Okay, bye-bye.
Thank you. Bye.